Have you ever had the blues?
Do you suffer from insomnia?
Depression?
Anxiety.
A gambling disorder?
Too much TV?
What about highly palatable foods and sweets?
Or TikTok.
It doesn't matter.
We can fix you. Therapy can take years to make a dent.
And who has the time?
No one. Hi, I'm Dr. Jenna Lee, postdoctoral research fellow.
I'm Lauren Raycraft, behavioral neuroscience PhD candidate.
I am Aura Pence, lab technician.
Hi, I'm just a med student.
What if there was a way we could activate certain brain regions in order to manipulate them and ultimately understand them?
But how is that even possible?
With laser beams.
Of course.
Laser beams? This just sounds like some sort of sci-fi fantasy.
Oh no. With optogenetic technology, we can make it a reality.
But how long would it take to even see any changes?
With optogenetics? Instantaneously.
Just take a look at this video, where we can activate reward circuits of the brain using
Lasers.
In the video, we saw a mouse moving freely around a behavioral box, with a tether connected to the top of the mouse's head from above. This tether was actually a thin fiber optic cable, which allows us to shine light directly into the mouse's brain. This is a technique called optogenetics. Optogenetics is a technique that allows neuroscientists to control the activity of genetically modified neurons using light. Specifically, light from a laser, which wasn't shown in our video, passes through a thin fiber optic cable, through an optrode or thin glass rod, directly into the mouse's brain. Here, it can interact with these genetically modified neurons to change their activity. Importantly, all of this is done while the mouse is awake and freely moving around. But how does this work inside the brain? In order to understand, we'll need to take a broader look at how the brain functions normally. You may already know that inside the brain there are billions of brain cells called neurons. Optogenetics works by changing the excitability of neurons. The excitability of a neuron determines how likely it is to fire what's called an action potential. Action potentials are signals that propagate or move between different neurons. Altogether, action potentials help to determine the neural activity, or the brain activity. In order for a neuron to send an action potential, it needs to become excited. However, neurons aren't excited all of the time, and in fact, neurons can change from being not excited to very excited in a really short amount of time. One of the things that helps to determine whether or not a neuron is excited is its cell membrane. The cell membrane is important because it separates what's inside the cell from what's in the space around it. Specifically, there are what are called ions, or positively and negatively charged particles, that exist inside and outside of the cell. The cell membrane controls the movement of these ions from inside and outside. In order for these ions to move into the cell, they must pass through the membrane. The movement of ions through the membrane creates what is known as the membrane potential. For our purposes, just think of the membrane potential as determining the excitability of the neuron, or in other words, determining the likelihood that this neuron will send an action potential. Ion channels are like little doors that exist in the cell membrane to help ions move inside and outside of the cell. Special ion channels, called opsins, are light sensitive. In other words, they respond to light by opening. This means that when light shines on the opsins, they open to allow more ions into the cell. Optogenetics uses opsins to make neurons sensitive to light. Once opsins have been added into neurons, neuroscientists can use a laser to essentially open and close these ion channels by turning the light on and off. In doing so, the neuroscientists are changing the excitability of the neuron in real time. As we saw in the video, a mouse used in an optogenetic experiment is able to move around freely and behave normally, even when connected to the optogenetic laser. When the laser is turned on, however, it activates cells in the mouse's brain and can then change their behavior. All of this happens in real time.
Now that we have our tools, how do we put this all together? First, let's summarize the elements in our biological toolbox. First, we have neurons connected to each other in the brain like a web. Second, we have the signal-carrying action potential of the neuron. Third, we have the neuron's excitability, which determines how easy it is to cause an action potential to fire. And finally, we have the neuron's genetic information, which determines what's released by a neuron and determines how excitable the neuron is. This final tool, the genetics of a neuron, is where the genetics in optogenetics comes from, and it's key to how the process works. Let's say there's a certain group of neurons that release a neurotransmitter which causes a happy feeling in mice, but those neurons aren't activating as much as they should be. This is how optogenetics could be used to cause those neurons to fire more. We can use the same technique to target neurons of any type, neurons for learning, neurons for sleeping, and countless other behaviors. First, scientists find a special mouse with a special chunk of genetic code found only in those groups of happy neurons. Then, we use very precise equipment and inject a virus into the mouse's brain.
A virus?
Yeah, it won't make the mouse sick, but it will seek out those happy neurons and their unique genetic code, and it will infect them by adding even more code. The new code allows the happy neurons to create opsin proteins and place them in the surface of the neuron. Opsin comes from the word optics, and these are the same sorts of proteins in our eyes that help us see light. This is also where the opto in optogenetics comes from. These opsins will make the happy neurons more excitable when certain wavelengths of light are shined onto them. This is where the lasers come in. We place an optrode in the brain, very close to the happy neurons, containing a thin glass rod that allows light to pass from a laser we control, and then through the skull and into the brain. When we turn on the laser, those happy neurons become more excitable, so they produce action potentials more often, and the mouse receives more of the happy feeling. Optogenetics allows us to observe changes in all sorts of animal behavior through changing the excitability of different groups of neurons in the brain.
A benefit of this technology is that we can activate cells in the brains in freely moving animals. That means we can study a wide range of behaviors. So for instance, at our laboratory at Michigan State University, we study the neurobiology of learning and memory. What that means is we study how we learn, what information is important, and how we'll behave in the future. So for instance, we want to know what areas of the brain are involved in deciding what environmental stimulus might affect our behavior, such as eating. So for instance, why McDonald's is so good at getting us to be driving down the road, see their golden arches, or hear their catchy jingle, and then turn in and end up getting an order of French fries. We can use optogenetics to turn on or turn off the areas of the brain that we think are involved in this behavior. We can also study other behaviors, such as drug addiction, so we can learn more about things like drug addiction and relapse. Other laboratories are using this technology for a therapeutic approach. For instance, we can look at modeling epilepsy by inducing seizures with optogenetics, then potentially create therapeutic approaches for people who suffer from epilepsy. So, what sounds like an idea out of science fiction is now being used in laboratories to study a wide range of behavior, from food intake to drug-seeking behaviors, as well as develop potential therapies.
You just learned how optogenetics uses genetics to manipulate the brain using light. We hope you found this experience enlightening.